So we now will have all three speakers on the panel. And I'm just going to start. Thanks very much for joining us, everyone. You could have yourself off mute, please, Dr. Hyman Petson as well. Okay, right. I'm going to start off by asking each of you the same sort of question. So the first one, I'll start with Dr. Hardiman. You've obviously got lots of projects going on. Is there any project in particular that is your pet project that you would like to share with us that you're currently working on in the clinic?
Yes, there is actually. So I completely agree with my two colleagues, and I think it's interesting that we all kind of took the same tack on our presentations. We didn't confer in advance, and we all think that genetic modelling, using genetics to model disease heterogeneity, the variability of the disease, and the really important value of biomarkers, and that the subject is the person, So Terry has done phenomenal and really important and interesting work over the years, and she's a giant in translational research, and her work has been really, really important, bringing it into the human domain, and the work that she's done on the mouse modelling and identifying this genetic variability is really, really important. What we're trying to do in our centre is really to think about the person with the disease as the exploratory subject, if you like, and that we apply the same rigour to humans with the condition as we would in a laboratory setting. And one of the things we've been really thinking about is there some way of getting at the variability of the disease using two modalities? And one is genomics and genetics and looking at ancestral origin, and that's really important. The other is looking at pathways within the brain. So one of the things that Terry said was that the human iPS cell, the induced pluripotent stem cell, doesn't necessarily, it recapitulates some of the disease, but our view, which we've evolved over the years, is that you probably need a functioning brain and a functioning nervous system to really be able to model this disease properly. And of course, it doesn't occur naturally in the animal world. It's a human disease. So what are the best ways to get at that? Now, there are two obvious ways, three really, what Angela's been talking about, the wet biomarkers. But there are imaging, so brain MRI scans, but they're very expensive and people have to lie down flat, and that can be very challenging. Or brainwave activity, and we've become really interested in looking at brainwave activity, the neurophysiology, the electrical patterns that are going on in our brains all the time. And could these be different in ALS, and can we capture them? And so the answer is yes, they are, and yes, we can. And not only can we capture them, but we can subdivide people with ALS into different subcategories just based on the patterns of disruption of their brain networks. So we've been working really hard on that over the last few years, and have really made some really interesting and I think quite important observations. Not only some work that we published in one of the big journals recently, a journal called Brain, showing this, but also we have some presentations at the symposium looking at whether this pattern change happens even before people develop the condition, if they have, for example, a C9orf72 variant, and the answer is yes, you can. So that's an area we're really interested in. We're interested in the signal patterns of the brain and the nervous system, and also how the brain talks to the muscles. So all of the different methodologies that we can use to measure this, and we think this is something that's going to be really important in the future, and that Angela will be using in the future in the studies that she is designing, and I think that'll get incorporated in the next 10 years into as a credible and valuable quantitative. In other words, we're not relying on my prowess as a clinician, but a data-driven measure of disease of how the disease is present, and a sort of a proximal evaluation of what's happening at a biological or neurobiological level within the neuroaxis.
Okay, thanks very much, Dr. Longerman. If I could pose the same question to Dr. Hyman-Petson.
Oh, well, let me first say what a privilege it is to be on the dais with Orla and Angela. These are two giants in ALS, and so I'm humbled by being here. I'm going to take a different tack. What Orla is doing is extremely important, and I wish I had something as fancy going on, but what one of my passions is, in addition to genetics and research and clinical research, is leveraging technology. And so the program that I'm excited about in the clinic is our program on brain-computer interfaces, which is really leveraging things that are available now and putting together systems for, I hope, under $5,000 that will use brain-computer interfaces to actually communicate, to run Google Home, and even a power wheelchair. And we've made some progress in starting to beta test systems where we're actually using things you can buy online now and putting them together and doing the software design to interface the different pieces of hardware. And so hopefully next year I'll be able to talk a little bit about some progress there. And I think some of our work was presented in posters at Allied Health. But that's my excitement, is to improve the quality of life and use the tools we have now to make life better. I think that's critical.
Okay, great. Thanks, Dr. Hyman-Petson. And the same question to Dr. Genge. Anything new, exciting that you're doing in your lab?
My lab is the clinical research unit, so it's not exactly a lab. This is really what I do. And it is great to share this with Orla and Terry. There are a couple of things. We are working on guidelines internationally for genetic testing, because if we don't know that you have a genetic disease, a form of ALS, then you can't be offered the therapies for the genetic form. So I think we've all firmly believe that we are still missing patients who could take advantage of some of these super advanced opportunities in therapy. And without a formal genetic testing guideline, many places cannot get access. So particularly if you work in a public health system, and that's a good part of the world, although it doesn't apply to Terry completely, although, Medicare and Medicaid and the VA would probably use these kinds of guidelines. There's a US-centric program happening, and we pull together the rest of the world. The aim is to have a tool that people can use to get the kind of support they need to get routine genetic testing in the ALS clinics. I say that's one really important. And the second really important piece, and it's nowhere again as groundbreaking as what Orla has been doing with her biomarker work, but it is really trying to get our outcome measures looked at differently. My new passion, which you'll hear about again next week, is to really take the ALS FRSR by both hands and shake it until it's better. It's not a bad thing, really, and it's a very useful tool in clinic, but we're asking it to do more than it can do in its current state. So we really need to sort of refine it to a one set of questions, properly translated, look at what's creating the variability, get rid of as much variability as we can, and really have it all under one umbrella. And I am basically going to be pushing these colleagues on the screen with me and a few others to truly help get this done, because my concern is if we don't do it, someone else will, and we may not agree with what a private enterprise could do to the ALS FRSR. So we're better to get it on it together. I had a patient advocacy group reach out and talk to me about this as well. So it's a really, really important thing for drug development.
Okay.
So I think those are my two biggest.
Great. Yeah, thanks for that, because that then brings us to a question that's come through from the public in the chat group. So basically, it's asking about the biomarkers. You know, what you were saying about the ALS FRSR. We're probably wanting to move away from that. So how far do you think we are from having a biomarker that, you know, not just FDA, but any drug regulatory body would actually accept as a surrogate endpoint? And also, what kinds of work needs to be done for that to be recognized by the different, you know, not just the FDA, but, you know, everywhere around the world, basically. And maybe we could start with, well, Dr. Gensch, do you want to take that one? Because that specifically touches on what you've just discussed.
So I will, and then I'll let the others have their important comments as well. I think we will know as some of the current trials finish and go to the regulatory bodies. Survival is the outcome measure that Europe in large part requires. And so we're not looking just for a replacement or an adjustment to ALS FRSR. And I'm hoping what we're moving towards is outcome measures and biomarkers that can be a proper combined assessment with survival and with a refined ALS FRS. And I think we have the opportunity to educate the regulators. The regulators don't want to block good drugs. They just want to be able to evaluate the drugs properly using endpoints that are objective, clear, and if we use biomarkers, that the biomarkers actually reflect the clinical disease. That's what they want. They're not the bad guys. They just need us to help them. And that's how I look at it. So I think we're not far. But I think we have to do it together.
Right. Dr. Hardiman, do you want to give us your feedback on that?
Yeah, I think there are two things. One, we have to decide what biomarkers do different things. There's a biomarker of disease onset. You know, there's a biomarker that says this is ALS versus this is not ALS. And then there's a biomarker, as Angela said, that should map onto what the drug is doing. And there's a biomarker then of what the disease is doing. And obviously they may be linked. My view is that I don't think that we're there yet with any of the biomarkers that we have. I think neurofilaments are very interesting and exciting, but I don't think we're there yet. Because neurofilaments are elevated in other diseases as well. The second thing is that I think we're going to probably have an array of biomarkers. I think ALS is more than one disease. In fact, I'm convinced of it. And I think that what we want to do is be able to segregate patients into the different subgroups. And I think we are going to need some kind of biomarker because clinically we're not able to do that. So genomics, can be a biomarker. So the work that Terry's doing is very important because that'll help us to segregate people. That's a biomarker of disease category. C9orf repeat expansion is a biomarker of disease subtype. So then there are many, many different types of wet biomarkers, but these are biochemical or protein signals within the blood or the spinal fluid, and they may be helpful. But I think we'll be looking at arrays. And in fact, the cartoon that I put up at the end, this precision ALS machine that we're building, the idea there is that we'll have some kind of methodology at scale to be able to segregate patients into subgroups using data-driven markers, both of disease onset and disease progression. So I don't think we're there yet. I think it'll be an array of different technologies. I think we're going to have to unpick the disease. We've done that to some extent with SOD1 and C9 and the other rarer types of genomics. But there's a lot more to do. ALS is associated with cognitive change in some people, but not in everybody. the types of behavioral change we see are different in different people as well. So there's a lot more work to do. Much of it, I think, is informed by the sort of work that Terry is doing, because I do think there are genomic signals. The heritability of ALS is about 0.5, so about half of the risk for developing ALS is in our genes. So we need to understand that.
Okay, and Dr. Hyman Petson, anything you want to add to that?
It's hard to add to that. I would simply.
Or anything new?
It's a, this was very thorough, and I think that one of the issues is going to be, as we do personalized medicine, I think underscoring the fact that the biomarkers may be different depending on the subgroup, and I'm wondering if, just to add to the mix, and Orla would probably agree with me, that sequencing of the whole genome for each individual is going to be an important part of the biomarkers, if you will. And each trial is going to have to be designed very thoughtfully about not only a biomarker of effect on disease, which would correlate, as Angela pointed out, with disease progression. You want a clinical biomarker or a biochemical biomarker, but also biomarkers of target engagement, and to interpret data, the genome, to know what influences other genes might have had on the effect. So
we need to be cognizant as well, given that we're in four corners of the world here, that we are very European ancestral population centric, and there are parts of the world where C9orf isn't important at all. For example, in Malaysia, SOD1 is important in Malaysia. So we need to be cognizant of this. There are other genes in other parts of the world. So the idea that we can target a gene and fix the disease, and the whole genome sequencing, we don't know enough about the genomics of outside of European populations, really. And so there's a lot of work to do there just to characterize what's important, what isn't, what's real and what isn't, what's actually pathologically important and what isn't. And that's a really important endeavor that's going to take some time, I think.
Yeah, absolutely. So that takes me to my question, because, I am coming from a resource limited setting. I don't know whether, this was going to bound to come up if you have me moderating. So how do we see, what we have now is really largely, as
that's my
as
cat.
a cat. Yeah, that's your cat. So Dr. Hardiman spoke about, how a lot of data is coming really from the Caucasian population. And we know that this is, multi-step. There are lots of different factors that are at play. So how can the ALS community, such as yourself, who have done all this amazing research, how can you bring in the rest of the world, people like myself, and also with Dr. Gensch, I think, we had one communication where, I tried to get a patient into the clinical trial. How do we get the clinical trials to, the rest of the world? So I, maybe we can start with maybe Dr. Gensch with that. Is there anything that you think that the community can do towards that?
Great question, Martina. We're actually looking much more frequently at getting trials outside of North America, and on a number of projects that I'm on, for exactly the reasons that we brought up. Sometimes the challenge is very silly, to use a normal word. The challenge is that the CRO finds it difficult to work in multiple time zones. Now, that's a really silly reason to choose a country or not choose it for an ALS clinical trial, but it's the reality. So a lot of it has to do with those of us, myself, Orla, Terry, others, who actually talk very directly with the pharma that are developing the drugs, and really encouraging them to look beyond their usual suspects, and get the trials into Asia, the Asia Pacific Rim in particular. The more I work with people who are not in North America and Western Europe, the more I realize that there's a tremendous potential and it's not that difficult. But really, a lot of the time, it's not a lack of interest on the sponsor side. It is the CRO and how they see the logistics. So we just really have to overcome a barrier that is a little bit unseen. That's really where I see the bigger barrier.
Yeah, I wanted to actually also ask Dr. Hardiman and Dr. Hyman Peterson. I mean, that is one aspect, which is the clinical trials, but there are also the other aspect, which is, you talk about precision medicine. And for that, you want to be having patients with, similar kind of like genetic predisposition, exposure to environmental factors. And to do that, you really need the rest of the world. So how would you see that? I mean, for instance, Project Mine. I mean, when I look at Project Mine, I see, there's obviously so much has come out of that, but there are a lot of countries missing there. So how do we move forward with this?
Yeah, so there are two parts to that, actually, Martina. Project Mine is a first world initiative in a way, if you think about it, because the Project Mine genome sequencing is paid for by the countries that donate the DNA. So we had to raise the money in Ireland to sequence the samples from Irish patients, and then that goes into the mix of Project Mine. So it is a problem. I think you're right. I think it's like the dementia project, the 1066 project. 66% of dementia work is epidemiologic work is done in 10% of the population of the world, and ALS is no different. And I think the Alliance is a fantastic vehicle to talk about this. Actually, I've had the privilege of engaging with colleagues in Latin America and in the Caribbean. So we received some funding from the American Centre for Disease Control to conduct an epidemiologic study and to start building collaborative work in Latin America. It's called the LANALS project. It's just finished. So we have a Latin American database now. And anybody from Latin America who's interested in engaging in ALS epidemiologic research, we'd be delighted to talk to them. So I think it's that sort of thing. It's working with centres and sites outside of the mainstream European and North American and Australian regions that have money, and trying to draw down resourcing from those parts of the world that are well resourced to support parts of the world that are not well resourced. So this is a global question. Of course, we are in the middle of a pandemic, and if you look at the map of vaccinations, it's no different. And so this is an global equity problem in general. It's not just relating to ALS, but I think we need to be mindful of this. It's a rare disease. We're a small group. We all know each other. This is something we should be able to do if we work collectively. So I think it is something that we need to pay attention to. We are excluding a large swathe of people in the world, and I don't think that's equitable.
Yeah, Dr. Hyman Peterson, anything you want to also comment on?
Well, I think all of the above, everything everybody said, and in addition, the development of the core groups like MEALS, NCALs, and PACTELs, these groups can also leverage each other and work together.
And don't forget LANALS. LANALS is the Latin American one now.
Yes.
Yeah.
Thank you, Orla. No, and so these groups should be having a discourse on how everybody can be elevated. To Orla's point that, we need to leverage all of the resources across the board, because what helps others is going to help us too. So what we learn from other populations.
Yeah, absolutely. And I think one of the things I wanted to also add is that you want everyone to be on the same page, collecting information in a similar way, but something that's realistic in the real world for countries that, that may not be able to do. For instance, the ankles prediction model, I mean, that's wonderful, but realistically, not everybody can do a C9 off 72, which is within that model, for instance. So I think, there must be ways where there is minimum information that will be useful, and it'll be nice for so for instance, for myself, because we're always constantly looking to see what's happening, but not everybody's doing that. So, we want to know that we're collecting the correct things so that we can actually collaborate with other people. So I think it'd be nice to have everybody kind of like come into this.
Martina, the reality of the prediction model is actually, it was built from European registers, and C9 is important in Europe.
Yeah.
In parts of the world where it's not, the genomic signature of other parts of the world may be the determinant there. So I think knowing your population is really important, and understanding what the factors are. We don't know enough about LS outside of European populations. It is probably more common in populations of European extraction. But the phenotype, the characteristics of the disease are probably different in other parts of the world. We just, we don't know a lot about, for example, the Indian subcontinent, and probably what does come out there does suggest that maybe different subtypes and phenotypes and possibly better outcomes in some cohorts within the Indian population subcontinent. So I think we need more data, and I think those data have to be generated locally so that the questions that we're asking are pertinent and relevant to the populations that we're studying locally.
Okay, thanks. Thank you so much for that. So I'm going to go back to, enough about me. Let's just get to some of the questions that are coming through. So this one is asking about the role of exercise and how it can help maintain function. Maybe we can ask Dr. Gensch on that one, based on your clinical expertise. So that's question number 6.
Great question. The person who's published on this is a neurologist colleague of ours, Nick Maragakis, out of Johns Hopkins, and my thinking and the thinking in the field has evolved over time. There was a time when we told everybody don't exercise at all. And as you know, that's almost impossible for patients with ALS, as many of them are quite active right prior to getting ALS, right? So my current recommendations are exercise as you would like, but monitor your own response to exercise. Don't take up weightlifting if you've never done weightlifting, and even if you have, be really cognizant of what you're doing to your muscles, but definitely keep your cardio and the exercise that you can do without causing yourself profound fatigue, which usually translates into muscle injury. You should do. That's my normal advice. I don't know if Orla or Terry tend to give other advice, but that's what I tell people.
Okay, so there's a question here about whether a person with the ALS could assess ALS-FRS by themselves, but I think that is correct. They can do that themselves. So that's fairly straightforward, that one. Yes. So there's there's a question which is, okay, I'm going to put it in a different way. So we've got, we've talked about biomarkers, we've talked about antisense oligonucleotides. These are things that are, antisense oligonucleotides are coming through, and that's going to be very helpful. And of course, you've got the stem cell also coming through, and there's been disappointment, obviously, with the neuron. But I think we want to just move forward and get your thoughts about how we can move forward with these two particular promising promising approach to management. So maybe we could start with Dr. Hyman Peterson. So we're talking about antisense oligonucleotides and also stem cell therapy with regards to management treatment in the future.
Well, I obviously from my talk, I think antisense is extremely exciting, and that strategy has been used in other diseases successfully, and it's moving forward in ALS. We just have to be cognizant of the caveats. But I think it's very exciting, and I think the way forward is clear. There's more work being done and more studies. And not only tofersen, there's an antisense directed at the extra DNA in C9 and that ataxin I mentioned. So these are studies that are ongoing, and I think that the horse is out of the barn, and we're going to see more and more of these, and in fact personalized, like with the FUS projects. Now, this was really personalized medicine for someone with a FUS mutation. So I think that, you know, these are going to be coming forward. Stem cells, we have to understand what they're doing, and I think that work is also ongoing to leverage the findings to date and to really know whether or not we're doing anything. And I think it's going to take some rehashing of data in the trials, not only neurone, but the other stem cell type of trials that have happened. And I don't think that we're that it's gone yet. I think that there will be more studies going forward as we understand better what happened in the ones that were done. We have to look where we've been so that we can look ahead and learn from what we've done. So I hope that answered the question.
Okay. Did anyone else want to add to what Dr. Hyman said? Not really. Okay, right. We'll move on to the next question. Is about Project Mind. Maybe Dr. Hardiman. Does it collect phenotypes of PALS as well?
Yes, it does.
It does, yeah. So that's quite straightforward. And then the other question is, of course, about whether the global pandemic experience may have altered the CRO mindset. I don't quite know how one answers that. Any thoughts on that? I think the CRO will just do whatever they want.
Well,
I don't know, Dr. Ganesh.
Orla, are you talking about Project Mind, or you feel you've said everything you want to say?
I mean, I think Project Mind does collect phenotypes.
Yeah.
I think Precision ALS, the new initiative, TRICALS, will be collecting a great deal more information about patients, and the plan would be that we'll link that up with Capture ALS, which is the Canadian project, and also the Answer ALS, which is US based, and some of the other large scale studies that are underway. So the idea will be that we will link up the genomic data with very detailed clinical information. But we collect, because Project Mine is linked, certainly in Europe, to our ALS registers. We have some core information on people, not probably as much as we should, but there is phenotypic data there. But we need more. We need a lot more to be able to segregate people into subtypes. Absolutely.
So that's great. No, CROs actually don't always have the final say, Nortina. So don't be so discouraged. You know, I'm an optimist. And
that wasn't my question, though.
I never give up. It's a part of the problem that I have. I'm getting treatment for it, but I think the biggest tool is what you're already doing in Asia with Pactals, and what Orla's referencing in Latin America. The biggest tool is creating a network and it doesn't have to be 100 sites. It can be one strong site per country, or two or three strong sites per country. It just, the impetus has to be creating that clinical trial site ability. And there's a few different ways to do that, because on our side, whether I'm talking as just a consultant and giving random advice, or whether I'm more involved in the program, we're really actively looking for opportunities to take new clinical trials into regions other than, and I'm not being pejorative, Terry, but other than the US. US has tons. But if you have a really strong site in Singapore or Malaysia, you don't need 100 sites in Singapore or Malaysia. You need one strong site. And that is enough. What?
We can have it in Malaysia.
Right. But that's enough to get a trial into a country. right?
Yeah, absolutely.
And that's kind of my thinking. And I think the best example I have is the growth of South Korea as a clinical trial site. And Dr. Kim's clinic is extremely important for us who are involved in any of the SOD1 programs, because there's a high incidence of SOD1 genetic mutations in South Korea, and he has developed a very strong clinical trial site. So even though the CROs may not bring up South Korea, I do every time, because I know that he can do exactly what is needed for the program, and his site can do it well. So that's all they want. They just don't like the inconvenience of having, but it's all about getting good patients and running a trial well. So
yeah.
Can I jump in there, because we were an underdeveloped country in Ireland for a long time, and until we joined the EU. Ireland had a terrible reputation of getting trials in. It still doesn't have a great reputation. By the way, a CRO is a clinical research organization. So it's the middle people who conduct the trial. The drug company will contract a center or a company that'll then help to drive the trial. And the key to building trials is, as Andrea said, it's to demonstrate excellence, and anybody can do that. You need somebody like Norteen in Malaysia, or Matthew Kiernan in Australia. Matthew's put Australia on the map, or Angela in Canada. I mean, the US has Niels, which was Terry was chair of Niels for a while. So you need some strong leadership, and you need excellence. And that doesn't have to be geographically anywhere. You can be, as long as you have strong leadership, very good data, and a demonstrable excellence. You can do this anywhere. It's about excellence, and that doesn't have to be. It doesn't have to be anywhere in particular. You just need people there to do it. That's all you need.
Yeah. Fantastic. Thank you so much for that. So I think before we end this, I ought to just go back to the first question on the board, which is really, if I could ask maybe Dr. Gensch first with regards to the MLX approval.
Sure. So MLX has submitted to Health Canada for approval. We are expecting the outcome from Health Canada as late as June of 2022, because they were given a regular review. However, we may get the result much sooner than that. And so they've been, Health Canada has accepted their application for approval for their drug in Canada on the basis of the Centaur data. Centaur was the name of the clinical trial that showed the benefit of AMX0035. And Health Canada is not at this point requiring the information from the Phoenix clinical trial, which is the one that's currently starting in Europe and the US. So we will know in the new year. The latest possible is June, but maybe much earlier than that.
Okay. And also, Dr. Hardiman, the question was also with regards to Ireland in terms of.
Yeah, so we're part of the European Union, unlike our nearest neighbour, unfortunately. I don't think Amylyx has actually submitted to the EMEA. But in any event, the trial was quite small. It was a small number of patients, and it was NSFRS slope. So we would like to see in Europe a larger trial. So it's not just the regulators. Those of us who live in Europe would like to, and the UK, would like to see a larger phase 3 trial with lasting much longer. The phase 2, what we would see as the outcome as being very. It's a study that looks very interesting, but we would like to see more data. So we are happy to progress to the Phoenix study and generate a large data set. So it's encouraging, but we would support the MEA in the request for a larger phase 3 clinical trial.
Okay, great, thanks. Okay, I think we better call it, put an end to this, but thank you so much to our 3 fantastic speakers. You know, it just so happens that, you know, for me, it's an all-female panel, so it's quite empowering, and I'm quite encouraged, and, you know, and I take heart all your advice with regards to how we can move forward anywhere around the world. You just need to just get in there and just continue to push on. And it's encouraging to hear that all of you will support anyone out there who's doing it for, you know, the greater good for the patients with ALS and MND. With that, I think, Kathy, do you want to come in? I'm not sure what we do now, but thank you to all the speakers. Yeah.
Thank you so much. I have extreme gratitude to express here. Nortina, thank you so much for moderating ALS MND Connect. Thank you so much to our esteemed panelists. That was a fantastic couple of hours. Hearing just a taste of what's going to happen at the research symposium next week in terms of what's on the horizon. I love the title of Dr. Hardiman's presentation, that we have reason for hope, and I feel a strong sense of that right now. I would especially like to thank our 3 ALS MND Connect sponsors, Brainstorm, Biogen, and Amylyx, for their support of the International Alliance to be able to bring ALS MND Connect to the whole world through our Alliance meeting, through our Allied Professionals Forum, and through Facebook Live. The recording will live on Facebook Live, and we will also be posting it to our YouTube channel shortly. I just wanted to say thank you for the very robust conversations you had about accessibility, about biomarkers, about genetics. Those are all conversations that are happening at the global level that are so important to our community. And especially about global collaboration as we all work towards our goal of a world free of ALS MND. And my last thank you goes to Dr. Felipe Ocampo, who started us off with his story of his journey and gave us, you know, the foundation of why this research that everyone is undertaking is so important. So thank you, everyone. And thank you again, Nortina, for your fantastic moderation of ALS MND Connect 2021.
My pleasure. All right. Thanks, Kathy, as well.